Biology 9. Okay, so we've gone through a lot of the organelles now, and now the question is, how do these organelles work together? So I want to think about protein production. So I want to look at the sort of the pathways that they move through. If you watched Biology 8, you may I did actually talk about this quite a bit. But for now, I'm just going to just get some more pictures on the go. And yeah. So we're going to draw part of a eukaryotic cell with necessary organelles. So let's draw the outline first of all. So we'll draw it as big as we can with the space we've got. We'll just do that. And like that. Okay. What should we do now? Now I'm going to fill it all in with this colour. I'm going to choose this colour, and we're going to make this the nucleus. So let's just draw a area, just try and do a relatively circular area, just about. It was a little bit sharp. Okay, that will do, I think. And we're going to fill that in with this colour as well, which hopefully looking good on that area. Yes, good. I just want to try and make these drawings as good as I can. Just do little touch-ups there. Okay, so that's going to be the nucleus. It's going to be just off. It's obviously it's just showing a certain section of the nucleus. Okay. So then what I want to do is I want to put the chromatin in the nucleus. So let's put the chromatin in there. We'll make these little dots in the nucleus. So just very lightly tap there. Okay. And then what I'll do is I'm going to draw the rest of it actually with, again, we're going to use a black outline. See how good we can get this. I'm going to really try to make it pretty good if I can. So we're going to try to draw let's do the rough endoplasmic reticulum. It's not going to be that easy to do it. This is perhaps a bit big. We'll see though. Okay. Let's just do something like that. Problem is, I can't actually. I probably, if I press undo now, that probably won't be a good sign. So I'm just going to. Alright, so that's going to have to be the rough endoplasmic reticulum. Okay, And let's draw the ribosomes on that. Okay. Just check it that nice and solid in there. So we've got the rough. And remember, of course, the smooth endoplasmic reticulum, or even just smooth endoplasmic reticulum, is not. That doesn't have ribosomes because it's used for lipids instead of for proteins. Okay. All right. So that's good. And now we want to make it go towards the Golgi apparatus. So that's going to also... Problem is I'm going to have to put the Golgi and then... Alright, so now we're going to put this little red arrow here. Okay, so we've got the red arrow, and this is going to go to the Golgi apparatus. Let's draw the Golgi apparatus. Could be easier, I suppose, if I just draw this, this, and then at any rate, at least they look quite different, which is kind of what I was going for. And we'll do some little vesicles, because obviously they're not actually connected, so they have to go through these vesicles. And believe I put the vesicles. Yeah. Okay. All right. So let's just fill that in a bit. See how we do with the fill tool here. Not bad. Not bad. Not bad. That didn't work. Then again, I haven't actually. I might be able to get. Yeah, that's going to be difficult, I think, to get. I might just go over that again. And I mean, I'll put yellow now. Okay, let me try and get this. So I'll just go like this. It's not too bad. Okay. So this is going to be the Golgi apparatus. If I can fill it, should be able to fill it. I just need to zoom in a little bit here. No, I didn't seem to get it, but I'm sure I can. Be very precise. It's very hard to get that bit. Let me just zoom in again. Yeah, I think that's about as good as we're going to get, I think. Okay, so there's the Golgi apparatus. And again, we're going to have some small vesicles moving outwards. So we're going to have another red arrow here. So it shouldn't be too difficult to use this diagram. I think it shows the key things. Okay, and these are carrying the little glycoproteins. across the membrane. So there we go, the little proteins there. Put a few more these membrane proteins. And yeah. So I actually think that's not turned out too bad. I might just put a little bit more, there, there, fill that in. Okay, put some ribosomes, just dot some ribosomes about as well. Okay, now let's label it with a colour that we haven't used, perhaps. I should just use black again. Should be alright. But that's going to be okay, I think. So what I might do is, okay, so let's just label it as best as we can. So we've got the nucleus here. Aria. There's the ribosomes on the aria. These are just 3 cytoplasmic ribosomes. 3 ribosomes. Probably should have gone a bit smaller with the pen here, actually. It's a bit late now. Vesicle. Okay. Yeah, these are actually little vesicles. I've got stuff in them. Okay, we're speaking about glycoproteins here. So they're the let's just change that 1 second. Okay. So membrane protein. Moves to plasma membrane. And I would mention it's a good idea to watch episode 8 because I did cover a lot of stuff in that. Stuff even from the should be useful from the first section. And then we have the plasma membrane. Okay, good. So that's basically, so we can see what's going on basically. So we've got proteins made at the ribosomes. So they're going to be here around the rough endoplasmic reticulum. And yeah, so they're made at the ribosomes. So they make proteins that are excreted or attached to the cell membrane. And the ribosomes in which are free here make proteins that stay within the cytoplasm. Now these proteins that are made here are processed, so they get sugar chains in here in the rough, but from the RER. Then once they've got that additional kind of information, they're sort of sent on their way like a little like the RER is like the big ship, and then these send little just sort of carriers over here to the Golgi apparatus. So more work with the sugar chains, and then they go back to the vesicles, and then they might, yeah. So and then from the Golgi apparatus, some of the vesicles are going to go to the plasma membrane, and some are just and they're secreted there, right? The secretory pathway, or they might just be transported about the cell. Okay, so that's a description of that. Now let's write it down. Seems like a good idea. So yeah. So let's just do this in a sort of stepwise way. So proteins are made at the ribosomes, as we know. I actually prefer using the paintbrush tool for this. I'm trying to remember which one it was now, though. Yeah, that one's not bad. So let's see if I can get a slightly better handwriting here, because I'd like to, if I can. Because I've noticed in certain episodes it's been a little bit inconsistent, and I'd like to try to improve it. Yeah, and you get more control with the paintbrush tool, I find. So proteins are made at the ribosomes. Okay. So yeah, so that's just a fact. So the ribosomes at the RER make proteins that are excreted or attached to the cell membrane. We always have to remember this is in 3D as well. It's the cell membrane. Okay. So yeah, so these are more. So it's not the same. Whereas free ribosomes. Yeah, I'm not going to delete every single little thing that I go wrong here. Whereas free ribosomes in the cytoplasm make proteins that stay in the cytoplasm. Okay. So there's different pathways for different proteins. That's really the key information there. Okay. So we spoke in Biology 8 about this. So we'll keep on going. So new proteins produced at the RER are folded and processed. So sugar chains. Added in the RER. Okay, so they're transported from the RER to the Golgi apparatus and vesicles, like I've mentioned. At the Golgi apparatus, there's more further processing So the sugar chains are either trimmed or more added. More added, okay. And then from there, it's more vesicles. So really, you can see the vesicles, little sort of small ships that transport things around. So yeah, proteins enter more vesicles. To be transported around the cell. So glycoproteins, for example, which are found in mucus. Move to the cell surface. And are secreted. Okay. So yeah, so that's going to be the first step here. Going to do some stuff on the cytoskeleton next time. So until then, thanks for watching.